Okay, thanks very much. So it's my pleasure to talk about a journey that I went through at the end of my PhD, trying to make a slightly novel data visualization, which involved balancing salience and completeness. So salience is really this. Oh, no, maybe it's not. Okay. So salience is basically how easy it is to differentiate things for our visual system and how obvious differences are when we look at an image. So we've had a lot of amazing talks today about interactive and reactive tools for doing fantastic complex data visualizations. But the reality for many people working in biological research is that the unit of currency is the paper, and the paper is published by a journal which requires static two-dimensional images to convey our results, and they have to be of set dimension and with set resolution. So I've maybe taking a bit of a step back into the Stone Ages in terms of the kind of cool stuff that has been presented today, but I think it's useful nonetheless to think about how we try and incorporate as much information as possible in a single image. So my PhD was working on this parasite called Giardia, which is a single-celled parasite that infests the human intestinal tract. And it's a pretty nasty little beast, causes diarrheal disease, and there's really not a lot of work that's been done on it at a kind of genomics level. The main drug that we use to treat Giardia is called Metronidazole, and that's one of only 2 drugs available to treat this parasite, which is concerning because there are some billion of the world's people infected with Giardia. So resistance to Metronidazole is really potentially a huge global health problem. There's a genetically related parasite called Trichomonas, which is also a human parasite of the reproductive tract. And these 2 parasites are both treatable with metronidazole, and there's also drug-resistant Trichomonas infections that are reported, and actually were reported during my PhD. So this is what kind of led to my approach to do something with these data sets, which are quite rare and quite expensive, comparatively speaking, in this really underfunded field of neglected tropical diseases. So when these parasites become resistant to drug, they either turn certain genes on or they turn them off. And we can measure how active a gene is by sequencing the RNA, which is the messenger that's transcribed from a particular gene. So the classic kind of differential transcription analysis looks like this, where you have each of these dots representing a gene, or more specifically, the amount of RNA that has been sequenced, that has been transcribed from a particular gene. And so on the x-axis, we have the average amount of gene of transcripts that are present in the cell, and on the y-axis, we have, in the resistant state, whether a particular gene is turned on or turned off. And so if the level of gene activation or suppression is significant, then it gets coloured in red. So we had this data. This was from a lab in New York for Trichomonas and from our lab with Giardia. And I was really interested to try and combine these data sets in some way that preserved as much of the quantitative information as possible. And so I needed some way to try and link up the genes so that they could be plotted on a single chart. And so there's this idea in biology that, related genetic species once had a single common ancestor, and that ancestor had a particular genome, and then as the 2 species diverge, they retain most of the same genes and there are slight differences. But you can go looking in the genomes of 2 related organisms for what we call functionally related genes, or orthologs. So we might be able to group the Giardia genes and the Trichomonas genes according to whether or not they're predicted to do a similar biological function in the cell, which is called an orthologous group of genes. And so we can do this with ortho-MCL. Now, so I went in and basically assigned the functional groups to these Giardia and Trichomonas genes, which were either turned on or turned off in the drug-resistant state, and came up with a table like this. And the sad reality is that I could probably have just submitted this to a journal and they would have taken it. Which, I mean, to be fair, they'd probably make it look a little bit more nice, but this is essentially what you see in so many papers, and it just really annoys the crap out of me. So we Because you need to go into the PDF file in most cases and try and turn that back into a table, then turn it into some kind of graphic if you want to interpret it. So, so what did I want to do? And I'm borrowing from the talk in the first session about the idea of design goals and then trying to assess, all right, how did you do at the end of the day? So I wanted to combine the results from differential RNA sequencing from these two parasites into a single chart, and I wanted to group the genes according to their functional group. To do a quantitative comparison that actually retained as much of the quantitative data as possible, and to display as much of the result as possible within reason, as well as highlighting the known resistance genes. So there's multiple layers of information that need to be encoded in a particular way. So this was the very first output from my attempt, where what you see is the different colours representing the functionally linked genes in either Trichomonas or Giardia, and whether or not they're turned on. If they're turned on, then they're above that dotted line, and if they're turned down, then they're below the line. And every point on this chart is a statistically significant result, so I've left out anything that is not significant in these data sets. And you know, it's actually not much better than that table because you can't differentiate the species. So the first thing to do might be to encode the species as shapes, because humans are quite good at differentiating shapes. So it's an obvious thing to do if you want to distinguish two groups. And so now Trichomonas are the triangles and Giardia are the genes are the circles. And it's very, very quickly apparent that there's a massive orthologous gene group that's basically composed entirely of Trichomonas genes. So there's one Giardia gene and 89 Trichomonas genes. So and this is really why I entitled my talk Balancing Completeness and Salience. These triangles down at the bottom of the chart are actually crowding out a lot of the rest of the data, but it's a very asymmetrical group because it's telling us about one Giardia gene and 89 Trichomonas genes. So especially because the Trichomonas genes are upregulated and downregulated, what can you really draw from that? I decided to actually cull them. So when you do that, you notice that your x-axis for your gene abundance jumps up to 3. So not only is this a massively redundant gene group for Trichomonas, but it's also hardly transcribed at all. So when I did this, I got something that is a little bit more informative, in my opinion, and allows us to see the data a bit more clearly. And importantly, in this chart, only the genes that are upregulated in both organisms or downregulated in both organisms are displayed. So the colours should group together above and below that dotted line. So that was okay. I wanted the readers to be actually able to tell what genes they were looking at, and so I wanted to label them. And when you go in and do that, it's, again, you really don't gain anything because it's massively overplotted. So you have to make another call. What am I going to leave on the chart? And I decided that I was going to leave the gene IDs for the genes that we know to be involved in resistance. And so when you do that, you can clean it up reasonably well. And you see that now you start to see what I was wanting to show, which is that the pairs of genes that are thought to do the same role in both organisms are transcribed in similar directions. So that's nice, but you know the labels are still messy and they're overlapping with the points. So then you go in and use a cool package called Geom Repel, I think it is, or GG Repel, to repel labels away from the data points so that they actually don't overlap. So this is starting to look a little bit better. And then the next thing I wanted to do is to test out a couple of different schemes. So what you could see before is just the default ggplot colours, and then this is, from what I understand, a colourblind friendly colour scheme, but maybe it isn't. And I think I certainly agree with what was said this morning. You know, this is 18 functional groups, and really you can use any colour palette that you want, but this is at the absolute limit of what can be differentiated with colours. And so, in my opinion, this is actually horribly ugly, and I would prefer to just stick with the standard ggplot colour scheme. But what was happening in the data is that the gene groups that tend to have similar magnitudes are actually grouping together, which means that the colours are very similar. So the last thing that I did is I went in and randomised the default colour scheme for ggplot so that that would decouple the magnitude of the gene transcription or regulation state from the hue that's used to encode it. So when you do that, you get this. And so now the genes which are upregulated are actually a bit easier to distinguish, in my opinion, and you have less of a situation where gene groups that are regulated in a similar way are also encoded in very similar colours. So in the context of the kind of limited time I had in the PhD, I thought that's probably a reasonably good way to preserve as much of the quantitative data as I can, whilst also allowing readers to directly compare how these two different drug-resistant species are regulating their genome. And so the last thing I did was just go in and actually annotate the functional gene groups which are known to be involved in drug resistance. And this has the somewhat unintended consequence of also making it a little bit easier for the reader to distinguish which related colours they're looking at on the plot. Because, for example, there's about 3 or 4 purples, but at least one of them is now taken away because you can say, okay, that purple corresponds to these chromate reductases. So, general principles, I didn't know these before I started, but I've kind of inferred them along the way. I used shape to differentiate large groups. And then for the gene ID, where there was about 10 different groups, shape differentiate a small number of groups, actually, because there's only really 2 species. Then the gene ID can differentiate about 10 with text, and then colour is really the only way to differentiate groups larger than about 10 that I could think of. And so I could find these little functions in R to help me along the way. So yeah, just to sum up with the design decisions, I was absolutely keen on trying to get all of the information into a single chart so readers could directly compare gene activation in these 2 species. But it meant that I had to cull one really big group of genes that was also highly asymmetrical. I ended up using the ggplot defaults because the colourblind friendly, or unfriendly as the case may be, palette was just too ugly for me. And I tried to compensate for the similarity of the colours by randomising them so we could differentiate the magnitude from the spectrum. But then you end up with a compromise where maybe the colours still aren't easy enough to discriminate, even though it's an aesthetically pleasing colour scheme. And finally, rather than being able to label all of the genes, I could only label a subset of them. And so that means that if the readers are really interested, they do need to go into supplementaries to find the other genes. So this is focusing on the negatives, but I think the positives are that it still conveys quite a lot of information and tries to make the most of what is a rare data set in my field. And this is the code, just ggplot with a couple of different features for anyone that's interested. And these are the guides that I've used along the way in research. I heard Martin Kraswinski mention before his column in Nature Methods Points of View. I would recommend to everyone. So thank you very much for your time. Any questions? Yeah, Robbie.
I really stumbled upon this web tool called Categorical. I don't know if you're...
Categorical?
Yes.
Yes, right, yeah.
It's basically like a web tool that is based on study.
Yeah.
And he basically tried to give you you just input a certain amount of classes that you want to invite.
Yeah.
And sort of spits you out a palette of color, depending on how many color, how pleasing you want them to be, how far apart you want them to be.
Yeah.
And like there are different parameters and stuff.
Nice.
Maybe like this.
Yeah.
Good.
It's great to hear about this kind of stuff. I think there's probably scope for another talk that just goes through all of the different tools that you can use to try and solve these problems. But yeah, that's a good one to know about, Categorical. Cool. Yeah, Errol.
Very general question.
Yeah.
I think publishing in STEM, one thing when it comes to sort of visualization that's unique to us,
Yeah.
or at least to our publishing overlords
Yeah.
and peer reviewers,
Yeah.
is we dislike originality.
Right, yeah.
So sort of push back and forth.
Yeah.
Oh, I've never seen this plot before. Please get rid of it.
Yeah, yeah.
So I just wonder if you had any thoughts or comments on originality versus conservatism.
Yeah. Well, I think, yeah, again, like Martin Krasvinsky's stuff is so impressive because he is trying to introduce these novel data visualizations into what can be quite an ossified field. So the idea of hive plots, that's really only been around for about 5 years, but it's really been like a revelation for getting away from those horrible hairball graphs. I think, this is a very basic general representation of data, but it's actually deceptively difficult to put a lot of information into a single scatter plot. So, but I guess I'm struggling to think of, and maybe this is just speaks to my lack of originality, of a novel type of data visualization that would do a better job, aside from what we all ultimately want, which is the ability to go in and interact with the plot, which would make life a lot easier in many cases. Yeah. Yeah. All right, I think I'm done. No? Okay, thanks a lot.